So again, good afternoon to all of you. I'm very happy to introduce today's speaker, Dr. P. Malati. Madam was our alumni and completed her UG, PG and PhD here. UG in Coimbatore, madam. UG Madurai, PG and PhD here. And I also had an opportunity to do PhD coursework with madam. It was really a wonderful period, madam, that one semester. Madam joined the ARS system during the year 1997, and completed nearly more than 26 years. Congratulations, madam, for completing 25 years. And Dr. Malati, madam, is concentrating her research on phylogenetics, diagnostics, and functional genomics of red rot pathogen. Madam is a specialist in red rot, not only these functional genomics and also other omics related to pathogenicity as well as antifungal genes of Coltotrichum falciparum. And Madam is also involved in developing the IDM strategies for the management of the red rot disease using physical, chemical as well as biological agents. And one important technology I want to say here, Madam has developed an instrument for SIP treatment, actually, which is using the osmotic pressure, madam. That one, vacuum. By creating vacuum, it's kind of our ordinary treaters, but by creating vacuum, if you treat these cells, thereby whatever material we are using for treating these cells, that will go inside, including bioagents, micronutrients, or whatever, or even chemical. So that is the advantage of infiltration technique for the treatment of sugarcane cells. And Madam approached us to concentrate or to do the same or use of this device in the other crops also, vegetatively propagated crops like turmeric, ginger, and also tapioca. Madam, for your information, that in tapioca we are getting good results. Dr. Ravichandran is conducting that experiment with salicylic acid as well as other biocontrol agents. So he has got very good results in the growth promotion as well as the disease resistance. So again, he is continuing in the next year also. So thereby, we will come out with a good device. That one can be used for all propagatively propagated material, vegetatively propagated material. Thank you, madam. And Madam has applied for the patent also for that infiltration device. Very soon, Madam going to get the patent for that. In addition to that, Madam also applied for another cytosolital gene-based specific for primer for Coltotrichum falciparum. So she has designed. So that is also under the way to get the patent. And I hope in near future this set treatment device will be of highly useful for all vegetatively propagated crop. And Madam has published nearly 87 research papers and more than 25 books and book chapters and several other technical papers as well as the conference papers and the general articles in the newspapers and popular articles. And Madam has guided 3 PhD and more than 15 MSc students from Bardar University as well as other universities. And she is the recipient of more than 13 awards and also more than 5 fellowships, especially from Biocontrol Advancement as well as a fellow of Applied Biotechnology. They are all worth mentioning. And similarly, other awards which include long-term overseas associateship in niche areas by DBT in South Korea, and also the very famous Professor M.J. Narasimhan Academic Merit Award, as well as the Sardar Lulu Memorial Award of IPS. And Madam is the recipient of Mule Deer Gold Medal during the year 2021, and she has been selected as Best Principal Scientist for the year 2021 in Sugarcane Breeding Institute of ICAR. And also, Madam got the Sri T.S. Venkatraman Award for outstanding contribution in sugarcane. So, with this small introduction, may I request Madam to proceed for the presentation. And not only in Madam title, if you have any doubt in sugarcane pathology, you can freely interact with the with our beloved Madam, and we can clarify your doubt. Thank you, Madam.
Going for presentation. First of all, I thank the academic committee, that is Dean Pages and others, for having given me this opportunity to uh, to conduct this public defense as well as to present this small topic. As I will be giving only outline of this work because I will not go in depth. Just if it is useful, it will be, because so far we are interacting only with the sugarcane people, not for pure pathologies means some conferences or something. Otherwise, we always interact only sugar factories, sugarcane for scientists like that. So this, since a pathology group, I am very much, this will be, I think, I feel it will be useful for interaction, and it will be helpful for interaction and also for further leading for any other fields also, like red rot, and you can take up in any pathogen and any host you can try. And first of all, and then our seniors and the most beloved, respected seniors as well as the teachers. Actually, the here all teachers, most of the super seniors are have gone. The immediate seniors are here. So Dr. Prabhakar sir also that immediate senior, and come teacher for us, or all seniors. So I thank everyone for having given me this opportunity to come over here to interact with you all. So if you have any clarification, you can interrupt. Thank you. Respected seniors, colleagues, and dear friends. Good afternoon to everyone. The topic for discussion is the application of OMICS tools to identify potential target sites for the management of red rot pathogen, the Colitoticum fulcatum in sugarcane. As we all know, always we used to think about conventional or molecular approach separately. But to bridge the both conventional and molecular approach, it's very rare to bridge that. So I could get some encouraging results to bridge the both approaches. So it will be some example. It will be an example for you for to conduct. Here I will be giving the outline of my research work targeted for this important pathogens, that is red rot. So as far as sugarcane is concerned, I need not give the importance of sugarcane. We all know about the sugarcane, and nowadays that is regarding sugar and also the allied products about that sugarcane. So biofuel, that is biodiesel, is a very important thing. So everybody knows the importance. And as far as sugarcane is concerned, red rot is the most important because it is the most destructive disease. And whatever we are developing the variety, we are mainly concentrating to develop a resistant variety only because of the disease. It is a challenge for breeders, both breeders as well as for pathologists. So it is and because it is responsible for elimination of several elite varieties under field conditions. Whenever, because in every year we are releasing the resistant cultivars only, but we are always, but it is succumb to pathogen, and a breakdown of resistance occurs very soon. It is due to the simultaneous development of virulence in the pathogen. So we always think about whenever we release the varieties, the simultaneously the present microflora, that is pathogen, that also develops and will attempt to establish in the host, newer host. So automatically it will try to adopt in the host, then it will develop a virulence. So then naturally it will result in the within 4 to 5 years the new variety will be naturally will lose its resistance. So automatically it will occur. So we thought of having, so we must understand very clear about the isolates, the set pathogen side, then only we can design the management strategy. So in that case, we were working on two different sides, one side on hosts also to identify and characterize the defense related genes for genetic engineering approach, and another side to identify and characterize pathogens related genes to tackle the pathogen and the field conditions as well as to under genetic engineering approach. Pathology section in sugarcane serves as the type culture selection for all over India. because every year we are going for survey and collecting the isolates and maintaining the isolates. More than 250 isolates we are having at present, and we have the well established preservation technique, and we have I have standardized the technique, so we are easy to maintain the all the isolates. Whenever we need the isolates without losing its originality, we can revive and study the any taken for any studies. So in that case, we have established the phylogenetic with conserved gene sequences for all the isolates, whatever we have isolated. Then based on the conserved gene sequences, we have developed, identified the specific primers and established the molecular diagnostics for diagnosing in the tissue as well as soil, and also for large scale application. Then third one is, then we applied it. Then after standardizing the diagnostics part, then we have gone for utilizing the omics tools like genomics and proteomics to characterize the pathogenesis related genes or proteins, as well as simultaneously the antifungal genes from antagonist also. Then finally we validated the candidate genes by applying the functional entering into the functional genomics, in which we have standardized the RNA approach as well as gene knockout as well as gene knockdown approaches. And then now we have a handful of pathogenesis related genes. We can use those genes for virulence determinants as virulence determinants, and also to tackle that genes, we are identifying the inhibitors for under field condition. This is the broad outline of this work. As far as Coltotic and Fulcatum is concerned, it has enormous pathogenic variability, phenotypic variability, that we can see this cultural variation based on cultural, we can see cultural variation, pathogenicity, and also vegetative compatibility, and also production of various metabolites like toxins, enzymes, and melanin. Then, although we have observed the enormous phenotypic variation, we could not find out any, we could not correlate phenotypic characters with any genotypic correlation. Since when we did the work with a group of isolates, every year many people have tried genetic variation, but they could not get any output because they have handled a small number of isolates, and they could not conclude any correlation with the phenotypic traits. So we have used isolates for covering 20 years of at least 15 years. Initially, we covered 15 years of isolates collected all over India, both from tropics as well as subtropics, and subjected these isolates to the conserved gene sequence analysis. First, we started with 5.8 as ITS, and here we could see these two major groups. We can see both the groups were differentiated only one base pair, and the third group is minor groups. It is only 5% isolates. This covers 95. So this 5% isolates were found to be least pathogenic, and they were culturally differentiated, and they were resembling the abroad the foreign isolates. So these are subtropical and tropical isolates were found to be virulent, and this isolates, third group isolates, this minor group isolates was found to be least virulent and were resembling to the foreign isolates. Then further, we confirmed this grouping with a multi-locus analysis with other conserved gene sequences like actin, calmodulin, beta-tubulin, GPDH, like that. And based on that, we found that here we used only representative isolates, and we found that there was no difference between group one, the movement of isolates between two groups was not significant. So we coupled in the same group, and the third group was different. So we could see that the one major group with the 95% isolates as virulent, and the Indian isolates were found to be virulent, while the least group 5% isolates were found to be least virulent in the Indian isolates, with resembling the foreign isolates. Then we confirmed this grouping with RAPD markers as well as ISR markers. Then while doing this, after studying all this genetic variation, we found that there is no significant difference among the genetic variation, and although we are observing the enormous phenotypic variation. Then we started standardizing the cultural adaptation technique, cultural adaptation under field conditions, and we found that then we did this cross inoculation. That is, virulent isolates on resistant cultivar, least virulent isolate on susceptible cultivar. In both the cases, when we isolate, when inoculate and re-isolate, inoculate, re-inoculate, re-isolate like that, we step by step, each generation, the pathogenicity was getting increased, as well as the cultural characters was improved, and then likewise, in both the incompatible interaction, we could see the improvement in the increase in the pathogenicity. So in that way, we could see that the pathogen is getting adapted to the newer cultivars, and the expression of and the pathogenicity is mainly due to the expression of pathogenis related genes, that is functional genes, not based on the conserved gene sequences. Although they are not significant, the difference, not showing the difference based on conserved gene sequences, they are showing their expression based on the functional genes. So we started to use these isolates for the characterizing the pathogens related genes or proteins for further confirmation. So from these cultures we have taken and studied under two approaches, that is under genomic approaches. First we started with the characterization of pathogenicity gene homologs. That is the gene homologs we have identified, and from the other coltotrichum species, about 28 homologs identified from other coltotrichum species were characterized in C. pulchatum, and they are found to be differentially expressed in response to virulence. Then second approach means we have selected one virulent, that is representative virulent and least virulent isolate, and we have gone for SSS, that is suppressive subtractive hybridization, and we could get the differentially expressed genes. Likewise, we have gone for protemic approach. Here also we have taken two different isolates, that is phylogenetically different isolates, and went for 2D gel electrophoresis, and we have characterized mycelial secretome and as well as proteins from host pathogen interaction, and based on differentially expressed and genes and proteins, we could identify the we could validate under during host pathogen interaction. Here you can see this is the virulent isolate from the major group. This is the least virulent isolate from the minor group. This is the solid medium. In the liquid medium, then we have extracted the DNA, RNA. We extract the DNA, RNA as well as protein for for further studies. Here during host pathogen interaction, we have inoculated virulent, least virulent, as well as in comparison with sterile water, that is mock inoculation. We extracted the samples in the inoculated position uniformly in all the cases on 5th and 10th day. Actually, for defense expression, we used to collect the sample within 24 hours or 48 hours. But for studying the pathogenicity genes, we have to extract the samples in later stages. Then only the pathogen expression will be there. So we were extracted the proteins from the inoculated portion. This is with respect to mycelial proteins. Initially, the samples were subjected for stage phase analysis. Then, after studying its purity, then we subjected for 2D electrophoresis. And here we have characterized only the differentially expressed proteins from virulent isolate. Likewise, we did in a secretome also. For in secretome protein also, we have differentially expressed protein from virulent isolates were characterized. Then this is with respect to host pathogen interaction. During host pathogen on 5th as well as 10th day we have studied. Then we subjected for 2D electrophoresis. Here also we have characterized the proteins from virulent isolate during here actually it's a ditrophic interaction, pathogen and host in a susceptible cultivar on 5th and 10th day. Then we characterize the protein. Then we have isolated and characterized the protein during tritrophic interaction. Here we have used the sugarcane tissue, pathogen, trichoderma, and then all the three. So during this tritrophic interaction, we have characterized the pathogens related proteins, because it is to cross-validate. Because during tritrophic interaction, the expression will be there, and during tritrophic interaction also the expression will be there. So in that we have to identify the candidate proteins, we have cross-validated, and here also we could get the same proteins as expressed in the ditrophic interaction. Then this is with respect to genomic approach, the characterization of pathogenicity gene homologs, because we designed the primers and amplified, and we have studied the expression analysis, and we have characterized in pulchatum. Then this is with respect to SSH approach. Here also we have extracted RNA and then subjected for cDNA, then for SSH approach, and here we could get very good results on the differentially expressed genes, and we found that more than 80% of the genes were found to be related to all Coleotrichum species, and one or two from Fusarium and other genera. Then based on these both approaches, we could get four important genes. That is bis1 family protein, that is 2-methyl esterase, and 3-isopropyl malate dehydrogenase. These four proteins were found to be our genes or proteins. We were cross-validated in transcriptome also. We have cross-validated, and we found that in the were highly useful for to measure the virulence, because every year we are isolating the isolates, and we are we are studying testing its pathogenicity under field conditions. But in field condition, if the conditions are not conducive, then we cannot differentiate the correct virulence of the isolate. But in by using this genes, it's very easy to monitor the virulence. And then the predominant PKS1, that is melanin synthesis genes, which was identified during that characterization of gene homologs, it was taken for further field studies. And then the dominant proteins HSP20 and cytochrome P450 were identified during ditrophic as well as tritrophic interaction, were taken for fungicidal management. And then remaining proteins, that mycelial secretome and many proteins from touring host pathogen were cross-validated with a SSH approach. So taking two approaches, we can cross-validate, and the results and the important genes or candidate genes can be taken further for any genetic engineering approach or for conventional studies. So these candidate genes, that are about 4 genes where we have characterized by the functionally validated by actually chitin synthase genes, we have validated by gene knockout approach, and the other 4 genes were chitin synthase by gene knockout, and the other 3 genes were by gene knockdown approach. We have standardized for the first time in Falcatum, and then we have published it also. Then regarding the potential, because from these two approaches we can identify the potential target sites for the fungicidal management, because the purpose is to identify the potential target sites is to identify any inhibitors or fungicides for the conventional management. In that way we could see that this PKS1 genes, PKS1 melanin biosynthesing genes are inhibited by the tricyclosol azole compounds. Mostly all the target sites are targeted by the azole fungicides. Here, this is tricyclosol is highly inhibitory to the melanin, and then cytochrome P450, it is highly inhibited by other azole fungicides, and hence here we have taken these two genes and demonstrated under field conditions. Here we can see at lower concentration of tricyclosol, it was able to inhibit the melanin synthesis as well as approsorin formation, and with increasing concentration as well as it inhibits the germination. It was correspondingly depicted in the leaf biopsy. Then we have inoculated in the standing cane also, and we have studied the expression of melanin synthesis genes, and we found that there was a highly suppression of genes in the combined inoculation, and it shows as like a resistant cultivar as compared to pathogen alone inoculated. Then this is with respect to cytochrome P450. This also has been cross-validated with the transcriptome analysis, and and finally we have taken this for field conditions. Here we can see, here we have taken, actually it is cytochrome P450 is a potential target site for azole fungicides, because that firm itself, they have given this as a potential target site for azole fungicides. So we have taken this one, and here under in vitro conditions, here we can see the propaganazole as well as tuberconazole, two fungicides, two azole fungicides we have used in comparison with the thiophanate methyl, which is different group. It is has different mode of action. That is, it has nucleic acid metabolic synthesis. It inhibits nucleic acid synthesis, but these two are targeting and the mitochondria, that is in cytochrome P450. So here we can see with the increasing concentration in both the fungicides, we can see reduction in the suppression of the inhibition or suppression of the pathogen growth, while it was not there up to 2.5 ppm in thiophanate methyl. Then correspondingly in qRT-PCR studies, we can see with the increasing concentration, there was a suppression of gene expression by the 2-azole fungicide and not by the thiophanate methyl. Then we validated this with the set treatment, and here also we have tried with different concentration of tibuconazole, and we found that the corresponding reduction in disease incidence as well as suppression of the target gene. Then we soil rinsed the cane with this tibuconazole, and on 5th and 10th day we found we extracted the sample, and we have subjected for qRT-PCR, and we found that there was no expression of gene in the tibuconazole treated plants. Based on all these things, then we have selected the azole fungicide for field application, because earlier we have identified thiophanate methyl as a effective fungicide for red rot management. Then we selected this azole fungicide also for individually as well as in combination with the thiophanate methyl. Simultaneously, when we are working on molecular side, in conventional side also, we have identified the effective molecules of fungicides as well as biocontrol agents. And after identifying the molecules, we could not recommend to the farmers for delivery of these molecules, because since the sugarcane is a voluminous planting material, it is difficult to treat in the fungicides or any biocontrol agents, because we in our research findings we found that at least overnight soaking is required, because the rind is there, so the effective infiltration was not there with a short period of soaking. So we found that at least overnight soaking, but here we can see it's very difficult to soak the sets in the for overnight. It requires a huge container, or and also the higher quantity of chemical, and finally after treatment it should be lodged into the soil. So everything this deleterious effects. So we found we thought of having some mechanized means of treatment to treat the sets with fungicides for field application, and we this is the prototype we developed, and based on that we have developed the bigger units, and we validated under the sugar factories from 2012 onwards, I think 13. Then here, this is with respect to thiophanate methyl treatment, set treatment, as well as soil rinsing with the Pseudomonas, drip delivery with Penibacillus or Pseudomonas fluorescens, and then now we have validated this azole fungicides individually as well as along with the thiophanate methyl, and we found it was very effective. Recently, the azoxystrobin is highly recommended for the sugarcane. So now we found this also is a inhibitor of cytochrome B1C complex. So finally, we found that all the azole fungicides are mainly targeting on mitochondria. So these found to be the efficacy of fungicide. We could confirm this based on the inhibition under gene level. Then now, based on our validation, now this in subtropical conditions, the red rot is the major limiting factor for our commercial, the wonder variety. Since we are in all the sugar factories, they have purchased this equipment, and they are uniformly they are treating with the fungicide for planting. So successfully, we are able to manage under field condition for red rot, and also other fungal diseases. Also, we are recommending this the treatment for the management of fungal diseases, because it is simple within 15 minutes for overnight soaking. When we compare with overnight soaking, it requires only 15 minutes of treatment, and so as far as handling, it is a simple as well as rapid and effective, economical, because repeated use you can take the same fungicide, lower concentration of fungicide we are using, and also repeated use we can have, and because of that it is eco-friendly and also practically feasible for farmers. Everybody, the farmers easily they are handling, and amenable for large scale application, and also able to deliver many inputs at a time. Not only fungicide, it will we can combine fungicide, insecticide, and nutrients, all the things at a time for the any type of sets. It need not be a bigger sets, and it may starts from single bud sets, and also for other vegetatively propagated crops we can recommend. And now, so we are effectively utilizing this technology for healthy nursery program as well as disease management. It's actually patented in filed patent in 2011, and it to be awarded, and it has been commercialized, and all over India, about more than 200 installation had been done,